So I'm going to talk a little bit today about water deficit stress in Thuja Plicata. My name is Don. A lot of the data collected here was done in association with Natural Resources Canada, and that was with Cosmin and Mike. We're both here today, as well as my supervisor, Jim, at SFU. I'm an MSc candidate at Simon Fraser University. So in the news recently, there's been some kind of wild headlines. These are sort of the most extreme ones that I could find. None of these are really, I would say, true, but Western red cedar is just one of many BC trees facing extinction is my favorite one, even though presentations we've seen today, as well as stuff that I'm going to talk about, would suggest that it's actually going to expand its range over time. There, however, is a fairly long history and a well-recorded history of drought issues in Western red cedar. There's a paper from 1988 here where they talk about how red cedar dies fairly quickly and it's sort of more often from drought than other species. And that's usually because it's a fairly shallow rooting tree and doesn't have some of the other effects that are sort of more typically drought resistant trees like Douglas fir have. So the effect of climate change on Thuja Plicata's distribution. So we have increased temperatures over time. Almost all models show that our temperature in BC is going to increase between sort of 1 and 2 degrees over the next several decades. There's going to be an increase in severe climatic events. So this is more droughts and water deficit. There's going to be an altered range. So we're probably going to lose some sites in which it currently grows. So those are the more southern or the drier sites. This map here is from a study that was done in 2014, in which they suggested that much of the southern range and places like southern Vancouver Island, where it's already sort of the drier end of its range, will lose. And it will expand more northward and sort of into the interior. This means that there's a need to understand adaptation and acclimation to water deficit for selecting adaptive provenances, so you can plant the best trees sort of where they're going to survive best, or like the ones that are best suited for that environment. This is sort of the first step towards that. So what is acclimation to water deficit? So in general, the rapid response is the signal perception. So this could be just like a lack of water, and that is associated with like ion channels, hormone responses. This then leads to signal transduction. So that's the MAPK cascades, plant hormone responses. Then that leads to transcription factor modification. So you end up with a whole bunch of transcription factors being expressed. And these in turn lead to responsive genes. So that's things like chaperonins or things that relate to cellular water homeostasis or cell growth and expansion, as well as amino acid and other osmolyte production, which then leads to physiological response. The slow response tends to be things like the common ones are, so like how often the plant will open its stomata, how thick the cuticle wax is. We're going to talk more specifically about mostly what happens in the stem, because that's where our data comes from for this section. Things like smaller tracheids or different cell wall thicknesses due to reduced water. So again, acclimation to water deficit is the modification of plant physiology and development. The fast response is the classic example, closing stomata, synthesizing osmolytes. Slow response would be things like making more roots or smaller leaves over time. In general, these are preceded by changes in gene expression. It is well studied in model plant species, which are usually a far cry away from the trees that we're talking about here. So things like Arabidopsis, the classic model organism for plants. The function of many of these genes and proteins, as well as the metabolites, are known. So our approach is to identify genes that respond to water deficit. A rapid response here, we did a study in which we looked at between 1 and 24 hours after exposure of roots, and by extension leaves, to osmotic stress, as well as a slow response where we looked at the sapwood of wild trees during the typically rainy period of June and the drier period of, we're going to call drought here, August. And then we identify the identification of responding genes provides insights into which processes are taking place at these times and what might be happening by extension later on due to these stresses. So the experimental setup for my first thing here, we took 4 seedlings and 3 treatments. Our control included no PEG, so PEG is polyethylene glycol, which is a molecule that structures water. We simulated approximately -1 megapascal. So even though these were grown hydroponically, they had their water potential that was available to the roots was approximately -1 or -2 pascals, and we call those low dose and high dose. We have 3 time points: 1 hour, 4 hours, and 24 hours after they were sort of placed in these treatments. I sampled their leaves and roots and then froze these samples immediately in liquid nitrogen and then performed RNA extractions using a standard CTAB protocol. We're only going to be talking about the roots here because the leaves haven't been sequenced yet. So in general, you select mRNA using poly-A selection, you create a sequencing library, we sequenced it, and then I performed transcriptome assemblies using Trinity and a variety of other programs. I then assessed the quality of these transcriptomes using Busco. I annotated them using UniProt or SwissProt more specifically, performed differential expression analysis, as well as gene ontology enrichment analysis. So in general, the transcriptomes produced were of high quality. So there's, Busco stands for Basic Universal Single Copy Ortholog, which is supposed to be sort of genes that are expressed in all plants. Most of them were found to be complete within our database. Only 10 were fragmented, which speaks to sort of the completeness of the transcripts. 91 were missing, although this isn't necessarily sort of unexpected, as it was made only using root tissue or wood tissue, depending on which one we're talking about. So after 1 hour, a large set of genes are upregulated in response to high dose of PEG, so the higher osmotic stress treatment that we had here. You can see this big cloud here of yellow. There's 2 of them. Those are the genes that are upregulated in response to our treatment. The low dose or low drought treatment and the control are much more similar in expression, share the same sort of normal, more normal expression pattern. So what sort of genes were upregulated in that yellow cloud there? Things like response to heat. A lot of it is things like chaperonin, so things that will immediately sort of try and allow for more normal function of your proteins when exposed to drought. A chaperonin is sort of a protein that is produced that helps restore proteins to their native or active functions. We also see things like osmolytes. So osmolytes often are carbohydrates or nitrogen containing organic compounds, so things like sucrose, sorbitol, mannitol, proteins like proteins as well as amino acids. Certain amino acids are commonly associated with drought responses or act as osmolytes. After 4 hours, our low dose of PEG osmotic stress results in a similar but weaker response in high drought. So we have a more similar expression pattern here now, where some of the same genes are upregulated in our low drought, and it is much more different now than the control than it was previously. In between our time points, we also see an increase in signaling. So between 1 and 4 hours, we see enrichment for genes that are involved in ethylene as well as ABA signaling. We're going to talk a little bit, and the expression pattern looks fairly similar where you at 24 hours, where you have high expression in the high drought, a slightly lower expression in the low drought, and then a different expression pattern in our control again. So at 4 hours, high drought activates additional genes. So we have the introduction of the TCA cycle or Krebs cycle, which provides malate, which is an osmolyte, as well as reducing power, NADPH, sorry, which is for metabolic processes, so producing other sort of compounds and secondary or specialized metabolites. You see an increase in purine metabolism, which is involved in producing antioxidant compounds, ATPase activity, which drives the active transport of ions. So that's sort of the second step that we were talking about. We also see an increase in signaling from hormone-specific genes, which I talked about on the last slide. Primarily ethylene and abscisic acid, as well as a little bit of jasmonic acid. A few genes from jasmonic acid are up there. So after 24 hours, The stress results in activation of processes such as amino acid biosynthesis and root development, as well as cell wall biosynthesis. So in general, you end up with thicker cell walls and more sort of cavation resistant tracheids in drought samples, so samples that have experienced stress. You also still have some of the protein folding stuff, so the chaperonins, as well as, you know, a variety of other things. But it's starting to try and sort of find more water at this time point, which is kind of interesting. Now that it's sort of started to calm down in comparison to the first time point where it was sort of an immediate response. So then to look at our slow response to water deficit, this is a site that's been managed for another reason, primarily looking at root rot. This is Mike and Cosman's site. There were several treatments originally on the site, stumped versus unstumped, and that was to look at the spread of root disease, as well as we looked at only our cedar trees, but there was two mixes there. So there was a straight cedar as well as a Douglas fir cedar, as well as random paired plots. And then we looked at differences in wood gene expression between the two time points historically associated with drying, June and August. In general, in Salmon Arm, which is where the site is, you have the most rain in June and the least in August. So those were sort of the reasons why we picked those time points. We have a very large number of replicates here. There's 104 total samples, and most of them are paired trees over our two time periods. We do see a huge number of genes upregulating in response to this sort of time difference in August relative to June. And the sort of breakdown of this is the majority of them are categories related to stress. So this huge brown thing here are all stress response genes, or categories relating to stress response. There's also the mobilization of carbohydrates, which is again talking about osmolytes and that sort of thing, as well as cell wall associated categories, because as I said, there is historically you end up with smaller tracheids and they're more embolism resistant, and that wood sort of is produced during the summer or the drying times. There's also the expression of secondary metabolites, things like terpenes and isoprenoids, as well as things like lignans are also produced during this time period. This is sort of just to show the hormone signaling. So from that subset, we found ABA, acetic acid activated signaling pathways, increased in August, which are these ones here, versus June. It's sort of to show that it's under stress, because ABA is the primary osmotic stress hormone in the literature. So most things, when they experience osmotic stress, produce ABA, which then leads to the downstream cascade and production of all the other compounds. So in general, what have we done? We've identified candidate gene processes for both rapid and slow stem responses. We also have genes that indicate ABA signaling, osmolyte biosynthesis, ion transport, cell wall biosynthesis, root development, and many others in sort of how Western red cedar responds to these treatments. We are looking at rapid leaf transcriptomes. We're currently getting ready to sequence those. And then we're also going to look at this in more detail, looking at specific genes and how they fit into pathways. And that's all for me today. Thank you.